Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT BET JRF entrance exam question paper conducted in the year 2016. I have divided into two parts and this is the first part. Coming to the first question. In an enzyme assay, the corrected absorbance reading obtained on addition of 100 microliter of enzyme extract is 0.025. Given that 1 unit of the enzyme is the amount of enzyme required to increase the absorbance by 0.001 units under assay conditions, the enzyme activity units per ml of the extract is. So 1 unit is the amount of enzyme required to increase absorbance by 0.001. So 0.025 is how many units? 0.025 divided by 0.01, that is 25 units. Enzyme activity is units divided by ml. Units is 25. 100 microliter, convert it into ml, it gives 0.1 ml. So 25 by 0.1 is 250. Then the cell-free extract prepared from E. coli cells overexpressing enzyme beta-glucosidase showed the activity of 1.5 units per ml. Protein concentration 2 mg per ml. The nickel NTA purified preparation showed the activity of 75 units per ml. Protein concentration 100 microgram per ml. Calculate the fold purification of the enzyme achieved. So fold purification is given by the formula final specific activity by initial specific activity. So initial specific activity is, that is before purification. So specific activity is activity units by total protein concentration in milligrams per ml. So 1.5 divided by 2, you will get 0.75. Then after purification, protein concentration is 100 microgram per ml. So convert it into mg per ml, so it becomes 0.1 mg per ml. Then specific activity is activity units by total protein concentration in milligrams per ml. So it is 750 units per ml. So fold purification is final specific activity by initial specific activity. You will get it as 1000. So the answer is option D, 1000. Then enzyme X showed its activity on substrate A, 375 units per ml. Substrate B 185 units per ml and substrate C 75 units per ml. With respect to the substrate A, the percent activities on substrate B and C are respectively. So with B with respect to A is 185 divided by 375 into 100, that is 49.3%, and C with respect to A is 75 divided by 375 into 100, that is 20%. So the answer here is option C, that is 49 and 20%. Then protein A from Pseudomonas putida contains 135 amino acids. The number of nucleotides present in the gene encoding the protein will be. So one amino acid is coded by 3 nucleotides. So 35 amino acids will be coded by 35 into 3, that is 405 nucleotides. And they are the coding regions without stop codon. So you have to add 3 to the final answer, that is 405 plus 3, that is 3 stop codons. Sorry, 1 stop codon will be 408 base pair. So the answer is option B. Then molarity of 50% NaCl solution. I have done this in previous question paper, I think 2013. The same question has been asked here, so I have solved it. You can go through that. It is 2.56. Then next one, the reaction velocity substrate versus substrate concentration profile for enzyme A using 1 microgram enzyme per assay. Similar experiment was carried out under identical condition except that the concentration of the enzyme used was 2 microgram per assay. Under these conditions, the kinetic constants. So when enzyme concentration changes, Km will remain the same but Vmax will change. So Km will not change while Vmax increases. Will remain the same but Vmax will increase. So answer is option C. Then which of the following bacterial cell suspension at A514 nanometer is equal to 0.2 will have maximum number of cells? So the one with smaller size. The one with smaller size will have the maximum number of cells. So that is mycoplasma is the one out of these options with the smallest living organism. Then two solutions of a substance, non-electrolyte, are mixed in the following manner. 480 ml of 1.5 molar first solution plus 520 ml of 1.2 molar second solution. What is the molarity of the final mixture? So M1V1 plus M2V2 is equal to M3 into V1 plus V2. Substitute the values and you will get 1.344 molar. Answer is option B. A 29% H2SO4 solution having a molarity of 3.60 would have a density of how much? Molecular weight of H2SO4 is 98. So it is given by the formula molarity is equal to percentage by weight into 10 into density of solution by molecular weight of solute. Substitute the values and then calculate density of solution, which is equal to 1.22. Calculate the turnover number for an enzyme if Km is 0.01 micromolar, Vmax is 10 micromolar per second, and E0 is the total enzyme concentration, 0.001. So Km, Vmax and E0 has been given, and we know that turnover number Kcat is equal to Vmax by concentration of E0. So 10 divided by 0.001, that will be 10 raised to 4. So 1 into 10 raised to 4 is the answer. Option A. Then consider three independently assorting gene pairs A, B, C, C. The probability of obtaining an offspring of this with two heterozygotes from parents that are they have given the genotype of the parents also. I am not reading that. You can read it on your own. So to get capital A, you have got the parents capital A, small a, capital A, capital A. They cross together and you will get capital A as half. Similarly, capital B, small b into capital B, small b. Capital B, small b into capital B, small b, you will get, you want in the offspring capital B, small b, so that you will get it as half. Similarly, capital C into capital C, small c. You want one small c and one capital C, that is also half. So what is the probability that you will get capital A, capital B, small b, capital C, small c? That is half into half into half, that is 1 by 8. So answer is option C. Given that husband and wife are both heterozygous for a recessive allele for albinism, if they have dizygotic twins, the probability that both the twins will have the same phenotype for pigmentation will be. So let the heterozygous husband and wife be given by capital A, small a. So upon crossing, you will get 3/4 normal individual and 1/4 will be affected. Probability that both the twins have the same phenotype. So probability that the first child is normal into probability that second child is normal plus probability that first is affected into probability that second is affected. So 3 by 4 into 3 by 4 plus 1 by 4 into 1 by 4. You will get answer as 5 by 8. Answer is option A. Then in a family with three children, what is the probability that 2 are boys and 1 is girls? So these are the favorable events, that is B, 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 G, G, G. It can all 3 girls or all 3 boys, or 2 boys, 1 girl, 1 boy in the first, then a girl, then a boy, a girl in the first, then a boy, then a boy. Then these are the other combinations. So out of 1, 2, 3, 4, 5, 6, 7, 8, 8, you will get 3 favorable events. So 3 by 8 is the probability. Answer is option C. Then LDL with cell surface receptors get internalized via clathrin mediated endocytosis. This process helps in maintaining the cholesterol LDL level in the plasma. However, in a disease known as familial hypercholesterolemia, a very high level of plasma cholesterol is found. This could be due to mutation in which of the following genes? Clathrin, no. LDL, no. LDL receptor, yes. Chances are there mutation in LDL receptor will result in very high level of plasma cholesterol in hypercholesterolemia. Then GFP when overexpressed in a cell remains mostly in the cytosol. A GFP construct is modified such that the resultant GFP protein will have a conjugated peptide proline, lysine, lysine, arginine, lysine, valine at its end terminus. If such a GFP construct is expressed in a cell, the modified GFP protein will be localized in the. So it is having lysine, lysine, that is arginine and lysine are more in the conjugated peptide. So it will be naturally be localized towards the nucleus. So answer is option C. Then next one. Succinate dehydrogenase converts succinate to fumarate. Which one of the following is true when the competitive inhibitor malonate is added in such a reaction? So it's a competitive inhibitor. So Vmax remains the same whereas Km increases. Answer is option C. Then digestion of a 5 kb linear DNA fragment with EcoR1 generates two fragments, 2 kb and 3 kb, while digestion of the same molecule with HIN3 yields 3 fragments, 0.7 kb, 3.5 kb and 0.8 kb. When the same DNA is digested with both the enzymes, it yields fragments of 0.7 kb, 1.3 kb, 2.2 kb and 0.8 kb. The right sequence of restriction sites in the DNA fragment is. So here EcoR1 it yields two fragments, that is 2 and 3. HIND3 gave 0.7, 3.5 and 0.8. Double digestion you got 0.7, 1.3, 2.2, 0.8. Now 0.7 and 1.3 they add up to 2 and 2.2 and 0.8 they add up to 3. So EcoR1 will be naturally in the middle and it will be flanked on either side by HIND3 restriction site. So the answer here is one EcoR1 site in between two HIN3 sites. So answer is option A. Then, in a mammalian cell, protein synthesis is regulated at the level of initiation by various kinases. During viral infection, which one of the following kinase is involved in regulating the step of formation of ELF2 GTP MET tRNAi ternary complex in the host? So it is protein kinase RNA dependent, PKR. Answer is option B. Which one of the following side chains of an amino acid is responsible for fluorescence in proteins? So it is the phenolic group which is responsible for fluorescence in protein. Answer is option C. Then DNA molecules labeled with N15 and N14 can be separated by. So they can be separated by density gradient ultracentrifugation. So based on the density, the two fragments will separate. Then match the chromatographic technique from group A with appropriate elution conditions given in group B. So chromatofocusing pH gradient, DEAE cephadox increasing NaCl gradient, G150 isocratic gradient and phenylcephalose decreasing NH4SO4 gradient. So the answer here will be option A, P2Q4R3S1. Then cytoskeletal organization of a cell is regulated by RAS GTPase. Answer is option A. In comparison to animals residing in warm climate, animals living in cold climate need thermal insulation. The cell membranes of the latter would be relatively higher content in unsaturated fatty acid. So in cold environment, the long chain fatty acid, they become stiff and become solid. So gaseous exchange will not be possible between the cells, and they have to make it semi-permeable. So in order to make it semi-permeable, they will have higher content of unsaturated fatty acid. Then 1 ml in glycolysis, the conversion of 1 mole of fructose 1,6-biphosphate to 2 mole of pyruvate result in the formation of how many NADH and ATP molecules? So they have asked not from glucose step, they have asked from fructose. So fructose 1,6-biphosphate by aldolase, it will get converted into, it will split into two. That is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. 2 NADH will be formed. Then this will give rise to 2,1,3-bis phosphoglycerate. ADP will be reduced to ATP and 2 molecules of 3-phosphoglycerate will be formed. Then 2-phosphoglycerate will be formed. Then phosphoenol pyruvate will be converted to pyruvate and ADP will get converted to ATP. So totally 2 NADH and 4 ATP molecules will be formed. So answer will be 2 mole of NADH and 4 mole of ATP. Answer is option D. In diabetic ketoacidosis, increase in which of the following would cause elevated production of ketone bodies? So diabetic ketoacidosis develops when your body doesn't have enough insulin to allow blood sugar into your cells for use as energy. Instead, your liver breaks down fat for fuel, a process that produces acids called as ketones. When too many ketones are produced too fast, they build up to dangerous levels in your body. So they have asked, in diabetic ketoacidosis, increase in which of the following would cause elevated production of ketone bodies? It is lipolysis. Lipolysis is breakdown of fat as fuel in the liver to produce ketone bodies, and when too many ketone bodies are present, they build up to dangerous levels in your body. So that is because of lipolysis. The next one. Which one of the following modes of inheritance is seen in cystic fibrosis? It is autosomal recessive. Answer is option A. Statins are very effective against hypercholesteremia, a major cause of atherosclerosis. These drugs reduce plasma cholesterol level by inhibiting the conversion of 3-hydroxy-3-methyl-glutaryl-CoA to mevalonate in the pathway for cholesterol biosynthesis. Option C. Measles, mumps, rubella, MMR combined vaccine represent which one of the following vaccine categories? It is live attenuated vaccine. The next one. A hemophilic man marries a normal woman. They have a daughter who does not show symptoms of hemophilia. If she marries a hemophilic man, what will be the probability of their son displaying symptoms of hemophilia? So a hemophilic man, that is this, marry a normal woman and they have a daughter who will be carrier for hemophilia. She will marry a hemophilic man, then there are 50% chances that the son, what is the probability of their son? Okay, I should have marked it over here. I made a mistake of marking over here. It in both the cases it is 50%. So there will be 50% chance that their son will display symptoms of hemophilia. So answer is option C. Then the conventional treatment for methanol toxicity is to administer ethanol. Which of the following explain the basis of this treatment? Ethanol acts as a competitive inhibitor to methanol. Answer is option A. What will be the angular velocity of a rotor in a centrifuge operating at 6000 revolutions per minute? So angular velocity is given by 2 pi rpm. So 2 into 3.14 into 6000 divided by 60, that is for converting it into second. It is radians per second angular velocity. So 6.20 into 100, that is 628 radians per second. Answer is option B. Then lysosomes of the cell were labeled with lysotracker red. Subsequently, these cells were infected with GFP transfected mycobacterium and observed under a fluorescent microscope. What will you observe? So what is lysosomes do? They engulf the pathogen and carry out lysis. An important strategy for intracellular survival of pathogenic mycobacteria relies on their capacity to resist delivery to lysosome. Instead, they survive within macrophage phagosome, or else I can say that mycobacterium can survive within the lysosome. So even though a pathogen, it can survive within the lysosome. So the GFP mycobacterium will be co-localized with lysotracker red labeled lysosomes. Answer is option A. Then next one. A linear DNA fragment which has 3 restriction sites for BAMH1 is labeled only at the 5' end. This DNA is partially digested with BAMH1 in such a way that all kinds of fragments are generated. Under these conditions, how many labeled and unlabeled fragments will be produced? Now, what is partial digestion? Restriction digest that has not been allowed to go to completion. That is, every time when you cut this DNA fragment with BAMH1, you need not get completely digested DNA fragment. You can get either 2 fragments or 1 fragment or no cutting at all, or even 3 fragments. So that is called as partial digestion. I have done it in the next slide. So this is BAMH1. It has got 3 restriction sites. That is 1, 2, 3, and this is the rest of the DNA portion. So if it is cut at only 1, 2 and 3, then you will get 1 which is labeled, 2, 3 and this portion, 4 which is not labeled. If it is between 1 and 2, it is cutting at 2 and 3 and 4. So you will get 1 and 2, then 3 and 4 unlabeled. Then if it is cutting between this portion, that is only 3 is digested, you will get 1, 2, 3 labeled and then 4. Or it is not at all cutting, means you will get the whole fragment. So you will get how many labeled? 1, 2, 3, 4. 4 labeled and 1, 2, 3, 4, 5, 6 unlabeled bands. Then next one. In the preparation of humanized antibody, part of the antibody molecule is taken from the mouse and the remaining is taken from the human through genetic engineering technique. Which one of the following statement is true for humanized antibody? So a type of antibody made in the laboratory by combining a human antibody with a small part of a mouse or rat monoclonal antibody is called humanized antibody. The mouse or rat part of the antibody binds to the target antigen, and the human part make it less likely to be destroyed by the body's immune system. So the mouse part will bind to the antigen. So that portion, so that is CDRs of the mouse IgG will be fused with framework region of human IgG. Now if it is framework, since it is framework region of human IgG, it will not be destroyed by the body's immune system. So the answer is option A. Then approximately how many molecules of carbon dioxide are produced daily by oxidative metabolism in adult human? Avogadro number is. So 1 mole of carbon dioxide is 1 mole of carbon into 2 moles of oxygen into Avogadro number, that is 1.2 into 10 raised to 25. Answer is option D. A 25-year-old man undertakes a prolonged fast for religious reasons. Which one of the following metabolites will be elevated in its blood plasma after 24 hours? So after fasting for around 24 hours, glycogen stores are depleted, causing the body to utilize energy stores from adipose tissue and protein stores. So next to be used, followed by glycogen depletion, is the triglyceride stores in adipose tissue. So triglycerides are separated into free fatty acids and glycerol that the liver respectively converts into ketone bodies and glucose. Ketone bodies are not metabolites. So answer here will be non-esterified fatty acid. Now what is non-esterified fatty acid? These are molecules released from triglycerides by the action of the enzyme lipase and are transported in the blood bound to albumin. They contribute only a small portion of body's fat but provide large part of body's energy. So they have asked for the metabolite and it is non-esterified fatty acid. Then which of the following is not a deficiency disorder? So it is polymyelitis. It is caused by poliovirus. It is not a deficiency disorder. Then electrophoresis of a purified protein in SDS phase in the presence of 2 mercaptoethanol yielded 2 bands of 35 kDa and 45 kDa. However, in a gel filtration chromatography, the same protein elutes as 80 kDa. What conclusion will you draw from the results? So gel filtration, it does not involve the use of any reducing agent like 2-mercaptoethanol or beta-mercaptoethanol. And beta-mercaptoethanol, as you all know, is involved in breaking the disulfide bridges. In gel filtration, since there is no treatment with any reducing agent, it results in separation of the native protein as such. That is why you got 80 kDa band. But SDS page with beta-mercaptoethanol treatment, it resulted in 2 bands which are of different sizes. That is 35 kDa and 45 kDa. If you add 35 plus 45, you will get 80 kDa. So 80 kDa is made up of two subunits which are different in size. So it is a heterodimer. So the protein is a heterodimer. Answer is option D. The next one. Which one of the following virus is the odd one out? So it is human immunodeficiency virus. Why? Because it is the only virus that doesn't cause cancer directly. Whereas all the other viruses, they are directly involved with cancer. The next one. DAPI 4,6-diamino-2-phenylindole is a fluorescent stain used for staining of. So it is used for staining of both DNA and RNA. So DAPI, it will also bind RNA, though it is not as strongly fluorescent. So the answer is option DNA and RNA, option B. Then during antigen presentation by antigen presenting cells, viral antigens are processed and presented to T cells in the context of MHC antigen. Viral antigens are processed. So they have asked for class 1. Okay, we'll see. By class 1 presentation. So that is the first option. So by proteasome and are presented along with MHC class 1 antigen. That's correct. So answer is option A. Then which of the following is not specifically protected by intellectual property? So intellectual property, it refers to creation of the mind, such as inventions, literary and artistic works, design and symbols, name and images. Type of intellectual property include copyright, patents, trademarks, industrial designs, geographical indication. So trade secrets is not considered as a intellectual property in India. There is no, so they have written it as there is no legislation that governs the protection of trade secrets in India. So the answer is option D, that is trade secrets. Then teichoic acid possibly plays a role in growth of bacterial cell by regulating the activity of. So teichoic acid assists in regulation of cell growth by limiting the ability of autolysine to break the beta-1,4 bond between N-acetylglucosamine and the N-acetylmuramic acid. So answer is autolysine. The fungus that causes athlete's foot it is trichophyton. Answer is option B. The following amino acid it is the structure of proline. So it is a hydrophobic amino acid. Answer is option A. Then a DNA fragment was cloned at the EcoR1 site of a plasmid vector that contained antibiotic resistant genes kanamycin and spectinomycin resistance. It was observed that all the positive clones containing the DNA fragment of insert grew on medium with kanamycin but not on media containing spectinomycin. In the absence of any other confounding factors, which of the following statement would explain this observation? So there is an vector which has got antibiotic resistance, kanamycin and spectinomycin. Now you have cloned it at the EcoR1 site of the plasmid vector that contained this antibiotic resistance gene. And it was observed that positive clones means those which contain the DNA of interest. They grew on medium with kanamycin but not on medium containing spectinomycin. So what explanation would you give for this? Now kanamycin, so you have to see where the EcoR1 site is located. So whether it is located in the kanamycin resistance gene or whether it is located within the spectinomycin resistance gene. So they got positive clones which grow on kanamycin. That doesn't mean kanamycin was harboring the EcoR1 restriction site, or else you could, when you insert the DNA in the kanamycin resistance gene, it would have inhibited the growth in kanamycin media. But here they do not grow on spectinomycin medium. So EcoR1 site is located within the spectinomycin gene. That is the correct explanation. Answer is option B. Then next one. A cDNA encoding a eukaryotic gene was ligated to an expression vector which was then introduced into E. coli for expression of the protein. The experiment resulted in poor expression of the protein. However, expression improved significantly in a Rosetta strain of E. coli, which demonstrate that the initial poor level of expression was due to. So a cDNA region from a eukaryotic gene was ligated, and it was ligated to an expression vector, and it was introduced. This expression vector was introduced into E. coli for production of the recombinant protein. But what happened? The experiment resulted in poor protein production. Now what did you do? You took the expression vector and but you used a different strain of E. coli. You took the expression vector and you introduced it into Rosetta strain of E. coli. Now why was there an initial poor level of expression in the first experiment? So the answer is codon bias. Now what is codon bias? Codon bias, the genetic codes of different organisms are often biased towards using one of the several codons that encode the same amino acid over the others. That is, a greater frequency of one will be found than expected by chance. So if one particular amino acid is coded by 6 different codons, the organism will have a bias towards using only one codon and not the others. That is called as codon bias. Now, why there was initial poor level expression in the first strain was because of codon bias. The next one. In Drosophila embryogenesis, the signal received from Bruchen proteins by follicle cells results in posteriorization of these cells. Knocking out of the gene for Bruchen in Drosophila will not result in the failure of accumulation of maternal mRNAs. Option A is right. Then a patient suffering from an infectious disease had high level of TNF alpha and interleukin 6. Assuming there are no other interaction, which T cell population is likely to dominate in his blood sample? So differentiation of Th1 is the combination of transforming growth factor and interleukin 6. Differentiation of Th17 cells take place through a combination of TGF beta and interleukin 6. But in addition, tumor necrosis factor alpha and interleukin 1 beta can further enhance T17 differentiation, but only in the presence of TGF beta and interleukin 6. So which T cell population is likely to dominate here? It is Th17. Answer is option B. Then next one. The niche of stem cell in human skin is? It is bulge region of the hair follicle. Answer is option B. Then a covalently closed circular DNA was in relaxed state in water at 30 degrees Celsius. What will happen to the supercoiling state of the molecule if water temperature increases to 60 degrees Celsius or decreases to 10 degrees Celsius? So increasing temperature enhances DNA flexibility effectively, leading to more compact folding of the double-stranded DNA chain. That is what happens in increasing temperature. So the DNA will undergo positive supercoiling at 60 degrees Celsius and will undergo negative supercoiling at 10 degrees Celsius. So the answer here is option C. Then acclimatization to high altitude in humans induces differentiation of. So low oxygen at high altitude lead to increased RBC, that is red blood cells. This allows more oxygen to be carried to the tissue. So it will induce differentiation of erythrocytes. Then next one. The protection against smallpox afforded by prior infection with cowpox represent. This was a repeated question. So immunization with cowpox lead to the production of antibodies capable of reacting with smallpox because the two viruses share identical or structurally similar determinants. This is called as antigenic cross-reactivity. Answer is option B. What is glycosuria? That is high amount of sugar in urine. Option D. Then 1,000 colorblind males underwent random mating with 1,000 normal females, neither carrier nor colorblind. After how many generations the allele frequency in both the sexes in the population will reach equilibrium? So these are the males and these are the females. This is the first generation. Now they undergo crossing. So there is a carrier and a normal male, and they will produce progenies. Out of this, the carrier female will again cross with the carrier male, not the carrier colorblind male. Then they will give rise to the third generation. Then comes the fourth generation. In fourth generation, you see that colorblind female is equal to colorblind male, 50-50. So they have asked when the allele frequency in both the sexes in the population will reach equilibrium is more than 3. Answer is option D. Then the mechanism that permits immunoglobulins to be synthesized in either a membrane bound or secreted form is. So alternative differential RNA processing has been found to control the alternative forms of expression of genes. Alternative RNA processing of the heavy chain immunoglobulin, that is mu gene, is an example. The mRNAs encoding the membrane associated, that is mu M, and secreted mu S form of heavy chain immunoglobulin IgM are alternatively processed from a common primary transcript containing signals for competing splice and cleavage polyadenylation reaction. If the transcript is cleaved and polyadenylated at the mu-S poly-A site, mu-S mRNA is produced. If it is spliced between C-mu4 and M1 splice junction, mu-M, that is the membrane bound mRNA is produced. So the answer here is differential RNA processing. Answer is option C. The next one, which of the following is not involved in antigen-antibody complexes? So covalent bonds are not involved, they are very strong bonds. So answer is option C. Ion uptake by the cells from extracellular environment is mediated through. So transferrin receptor. It is transferrin receptor that is involved in ion uptake from extracellular environment. Then, which of the following viruses causes liver cancer? So, hepatitis B also will cause, hepatitis C also will cause. So, both the answers are correct. Option C and D. Then, which one of the following enzyme is an established intracellular antioxidant? It is superoxide dismutase. Answer is option C. A given cytokine has different biological effects on different target cell. This is known as, it is called pleiotrophy, because it is the ability of a cytokine to exert many different types of responses. So it is called as pleiotrophy. Then influenza virus contain 11 proteins, of which the two major glycoproteins are H and N. What do the H and N stand for? So H stand for hemagglutinin and N stand for neuraminidase. Answer is option B. Inosine in the tRNA anticodon will base pair with all except which of the following bases in the codon of mRNA. So it will not base pair with guanine. Answer is option D. Then, in electrophoresis, if the buffer pH is above isoelectric point of the protein will. So, at solution pH that is above Pi, the surface of the protein will be negatively charged and they will migrate towards the anode. So, answer is option A. Circulating testosterone is mostly present in bound form. Which of the following proteins bind weakly to the major amount of circulating testosterone? So it is albumin. Answer is option D. Then in the blood ABO blood group system in humans, alleles IA and IB are co-dominant and both are dominant to the allele small I. If a type B genotype, that is IBI, women marries a type A genotype IAI, the probable children to the couple would be of the type. So this is the cross that is obtained and you will get AB, B, A and O. So the answer here is option A.